Thank you, Nick, and thank you, everybody. Thank you guys for participating. So we'll keep it short and lively like we talked about. Why don't we start by just introducing yourself in a couple of lines and what you're most excited about at your workplace right now.
Hi, I'm Jen Ibrahim. I'm the head of the medical function in rare disease for the U.S. at Sanofi. I'm a medical geneticist by training, and so this is really, I'm very excited to be experiencing this next evolution in newborn screening. All of these conversations take me back to all those 2 p.m. Friday calls that I got from the state of New Jersey and how different those calls may be in the future.
Fantastic. Thanks, Paul.
Hi, Paul Kruszka, Chief Medical Officer at GeneDx. First, I'd like to thank Robert, David, and of course you, Nick. I know you're doing all the work for organizing this great conference. Again, this year, I was there last year and really excited. What I'm really excited about is really the diagnostic odyssey. I'm still a practicing clinical geneticist at Children's National in DC, and the possibilities here for newborn screening to eliminate that is tremendously exciting. I think I've used all my political capital actually at my company trying to promote newborn screening, but it's something I'm very passionate about.
Thank you, and likewise, it's very exciting to be here as well in the second year of the ICoNS meeting. I'm Vanessa Almendro. I'm the head for innovation at Danaher Corporation. I bring, I guess, a different perspective. We do not develop drugs. Danaher Corporation develops the technologies that many other pharmaceutical companies use to manufacture drugs. And our connection with newborn sequencing is that we are really thinking about that future where we will be identifying patients at birth. We need to be able to develop the right drug at the right quality for the right patient, and we still need to develop many technologies that will allow us to act on those rare disorders when they are diagnosed.
Okay, fantastic. So in a few minutes, we're going to open up the panel to questions. So please be thinking and prepping your questions for the panel. We got a great sort of diversity of points of views here. So a lot of this has the assumption that we're going to have lots of medications that will treat rare diseases. And those assumptions rest on a couple things: that they'll continue to be developed, that they'll be affordable, that they will be embraced, and that somehow there'll be a window of opportunity for treating people early once we find that we probably won't want to treat them just because they're carrying the variant, but we'll want to treat them before they develop the full-blown disease. How do you, as someone who works in pharma, how do you think about that? I've been dying to ask this question more frequently, so here you are. I get to put you on the spot.
Yeah, this is a huge area of interest for us, right? And many people in this audience have been involved in very long-standing natural history studies that can be a little bit painful to get done, and we depend so heavily on that information, knowing at the same time that it's not perfect. You know, who's able to enroll in those studies? What biases that we don't recognize do we bring to the design of those studies that maybe are excluding certain populations? So there is a bit of a balance, right? We're trying to go forward, hoping that we have the right information, sometimes discovering partway through that we didn't exactly have the right information. But I think we're doing a much better job of recognizing how public-private partnerships and, not to be forgotten, the voice of the patient, how much all of that can be much better incorporated into our work.
But do you guys sit I'm trying to ask behind the scenes questions now. Do you guys sit in a room somewhere and you say, okay, for these 100 conditions, we're going to plot out what would be the orthogonal testing that has to take place with a at-risk child? And then this is probably the place where we would intervene with a drug? Do you get that granular already?
Yes, not me personally, but certainly the research and development arm. They do, sort of condition by condition, try to take a broad look at how does this get diagnosed? What is the orthogonal testing? And I think one of the interesting things that's happened during my time at Sanofi in the last 10 years has been that the R&D group, and I think this has been true in other companies, sort of exist in their own world and are making decisions without the input from the medical team, which is the arm of the company that interacts with all of you and is getting insights about, what is it like to see one of these patients in the clinic. Most recently had that experience being able to help inform our research and development group about products that they're developing for OI and for achondroplasia. It's very difficult to do that without a clinician sitting at the table. So, that's one of the things that's changing.
Yeah, that's so interesting, isn't it? Because there's sort of an assumption that the echocardiogram or the EKG or the metabolic test that you need to follow that, presumably child with, is going to somehow get done, get paid for, and then that there will be an agreed upon threshold for when that child warrants treatment. So that's a lot of standardization that's not in place.
It's a lot, and it is one of the reasons that we do things like convene advisory boards to try to get that kind of information. At the same time, we want to, one of the lines that we, or the tightropes that we walk, is we want to fund and make possible that kind of work without inappropriately influencing it,
right.
Influencing the subsequent publication. But we see that as a way that we could help.
I mean, just thinking out loud, I wonder if that's something ICoNS could help with, trying to get some kind of consensus on if you have at-risk children in a particular category, what would be a general consensus on when their orthogonal testing pushes them into a treatable category or not. Any drug, even if it's not expensive, would have side effects. You wouldn't want to apply it to a kid who was showing perhaps nothing, right? But how many symptoms or phenotypes or metabolic abnormalities or X-ray findings do you have to have before you say, okay, I want to start treating that kid?
And as we heard earlier, that conversation that happened through the Liberty Fund sounded really, really fascinating. When those kinds of decisions are being made, what are the biases that you're bringing to the table? What I may think is an unacceptable burden of disease may be completely different from what someone else thinks.
Right, and as Natasha and Aaron and Holly pointed out earlier, equity often gets, the universality of blood spot newborn screening often gets touted as an equitable first step in most societies. But from there, even the conventional newborn screening process quickly falls off in terms of access to specialized care. So we all could set some kind of standard, and that might be great. But the society that we're in will determine what kind of access people have to that follow-up testing. And one of the things I just so admire from you guys from Genomics England is how you explicitly said, that the list has to be for things that are available for treatment. Now, you'll have your own biases in who goes for treatment, who follows up, who wants to be followed up upon, I suppose. But boy, by putting it that way, you've at least got a great start on that. So moving on to Paul. Paul, one of the points we've made several times this morning has been that there is this sort of newborn screening ecosystem that we're all pretty familiar with and that we all reference quite a lot in these discussions. Because we're using the word screening, because we're using the word newborns, we're referencing the state or national semi-mandatory, semi-unconsented newborn screening programs. But we've mentioned several times in the previous panel and my opening remarks, that's not the only way to look at this. You're in a diagnostic company. Is there a different way of looking at this that actually is more in line with, medical care and diagnostic care and what pediatricians do with their patients in their offices, rather than what a state or national public health service could implement.
Yeah, so those are some good points. I work, like I said, for GeneDx. We're a commercial laboratory in North America, focusing on exomes and genomes, rare disease, and the clinicians here send us samples on very sick children, and they have a diagnosis before. They're not presumably healthy. They're unhealthy. They have a diagnosis, and we are a diagnostic lab. That's what we see. That's what we do. The ascertainment bias is there. We have 100% complete penetrance in the samples that come to us, right? Now, we are supporting Guardian with Dr. Wendy Chung in the RTI early check study, Don and Holly somewhere up there. Those are totally different. Totally different. Screening for us as a company, totally different. And I have to remind people within my own company, we have to divide that line. This is screening, this is diagnosis. And we do a lot of rapid testing in the NICU as part of our commercial business. And so that really confuses the story. Rapid testing in the NICU, again, these are kids with seizures, hypotonia, feeding difficulties. So it really muddies the water. So we really have to, in my company, draw that line. Screening, we don't have a screening commercial product in my company. So this is new to us. If this ever becomes a product, at this point what we're doing is we're supporting 2 research projects to really answer all these questions. As Sir Michael Richard said this morning, the 2 things, the benefits have to outweigh the risk at a reasonable cost. I think that's what we're trying to show in these research projects. My concern with this, not really a concern, but my being my most pessimistic self, is that there is such an educational gap in our community, in the physician. I'm a clinical geneticist, in the physician community, and I was at the National Institutes of Health for over a decade, just working on my 3 or 4 conditions. I really didn't look past the walls of the NIH, but now I've been at GeneDx 2.5 years. I see, at least in North America, there is an incredible gap in education. What is an exome? What's the difference between an exome and a microarray? What's the difference between an exome and a genome? We're barely even scraping the surface of the diagnostic kids out there that should be getting testing and evaluations. But my most pessimistic self is how would we educate people? Let's say we make this a commercial project, which I think you're alluding to, Robert. How are we going to educate people, physicians, the community, that if this is a good idea, benefits outweigh the risk and it's a reasonable cost, that it's a good idea to get your infant tested with genome sequencing in some type of panel or X amount of genes. I think that's the first big hurdle. And going back to the diagnostic business, let's just look at the NICU, for example. We have everything on our side as a commercial lab, I would say, and as an ecosystem. We've got the medical literature in the NICU, thanks to a large part Stephen Kingsmore, his work at Rady Children's, and others. I must acknowledge others. We have guidelines. Well, I guess we don't have guidelines in NICU, but we have ACMG guidelines for other things. We have the payers. The Medicaid is coming around state by state in the NICU. The one thing we don't have is provider behavior.
Right, and the provider behavior may be the last thing to switch. And so part of the question I would ask you, Vanessa, as we start with you, is I think all of us are parents, or we know parents, and are parents going to be the place where to drive this if providers are not? And the second thing, Vanessa, I'd like to ask you about is trust. We've heard so much about distrust of people who are going to sell my data, distrust of people going to share my data. You're a corporation. Do people trust the government to hold their baby's data? Do people trust a corporation to hold their baby's data? Vanessa, what are your thoughts on some of this? And then I want to ask you a little bit about Danaher as well. But
yeah, no, that's it's an interesting question. I've heard, again, it's not my field of expertise, as you know, but I've heard when we've been talking with some of our partners, mixed feelings about trust and how to handle the data. I think it's as you were pointing on the education of the physicians, it's also an education on the parents. And one of the common answers that we heard is the lack of trust, just because they don't know what's the actionability, what's going to come next. It's not really where the data sits, it's where do I need to do this test? Why am I going to get all this data if I cannot act on this data? I've heard even comments about, from an insurance perspective, is this access to newborn sequencing exposing really what the underlying genome of my baby is and the risk for adult onset diseases going to change anything in the future about access to healthcare, or even from an insurance perspective? So that's challenging.
That's a major question that comes up over and over, and we'll be having an ethics. It's fascinating because, as so many people in this audience and others have pointed out, when you have a family with a rare disease or any kind of disease, they are the strongest advocates for, wait, let's get this done. I wish I had gotten this done. Mm-hmm. But the general public, with the assumption of good health, is often slow to embrace the idea of discovering a piece of probabilistic bad news. But from the Danaher perspective, how do you see this whole world, this upstream downstream world, and how do you think a company like Danaher can participate?
Yeah, so actually I'm going to continue to provide an anchor thought here on the Radius Hospital, right? We've been talking with one of the pediatricians there in the NIQ, and she said she's using already rapid genome sequencing to diagnose the babies. And she's a very sophisticated doctor that knows a lot about genomics, to the point about education. But she told me, Vanessa, what I don't know what to do is what to do next. The only thing I can do is to go to the father or the parents and tell them, well, I have a diagnosis for your kid, but the prognosis of those kids will depend on what we can do after. And there is really not too much that we can do when we identified some of these genetic disorders. I want to point towards some statistics that can frame a little bit how we are thinking about it. There are 30 million people, at least in the US, that are expected to have rare disorders. Half of those are children, and we were checking in our prior presentation, most of these kids will have neurological disorders, where early intervention is critical to really have a clinical impact. So now let's put us again in this future where we might be able to identify these kids at birth. What we want is doctors like the one I was talking to not mention the name, that they can know what to do with the data, where to go. And most of these are going to be rare disorders. So we need to really think about how do we now enable the development of treatments for personalized mutations. And that comes into technologies. That's the angle I'm trying to connect with Danaher, right? Again, we have many customers, many partners that are actually the drug development companies, which is fantastic. We have huge bioreactors. We do tons of technologies for cell therapy manufacturing, viral vector manufacturing, you name it. Danaher has a very strong network. But it's high volume. That's what we have been doing for the past many years, because that's what the industry needed. Now, what we are seeing is that more and more and more, we need to take the technological expertise that we have in-house to start thinking, how do we get these workflows for drug development and collapse them and make them smaller, still at the same quality, so that we can start enabling a future where hopefully we will identify the patients at the right time, hopefully working in novel regulatory standards, and we know how Peter Marks now is launching many initiatives to standardize regulatory as well, where we get to the point where a baby is diagnosed and we can press a button, that sequence has to go somewhere where we can then manufacture the treatment, right? So a little bit where Danaher is going right now is doing a lot of research and investments in developing the novel technologies that will help us to collapse manufacturing workflows.
I like to tell this story. you're a cardiologist and some wise guy like David or me identifies a variant in your patient or a patient that comes to you without a family history and with a sudden cardiac death mutation. And so you've got a couple of choices there. You could do nothing. You could do an EKG every year or every month. You could do an exercise EKG or some fancy electrophysiological studies. You could just skip the whole thing and put in an implantable defibrillator. You got a lot of choices and very, very little guidance. We can automate an awful lot of what we're doing here, but it's very hard to automate clinical judgment. And it's very slow to get all of the academic poobahs to sit down and try to come up with academic consensus statements that would cover not just every general case like sudden cardiac death, but every gene or every variant in every gene, which is the granularity level that many of us clinicians are actually working in. So this is a tough problem. Any thoughts from any of you on how do we scale clinical judgment? We'll open this up to you guys for creative thoughts in a moment. And get your questions ready, because we're going to switch to the audience questions in just a second.
I mean, I would say it's going to be tough. There's 7,000 rare diseases. ACMG has these things called ACT sheets, which are really nice, mainly inborn errors in metabolism, tells you what to do. But to have 7,000 ACT sheets is not going to happen. And you know, I hope, you know, we go to medical school to learn the art of medicine, and we're going to have to figure out, well, probably, you know, maybe it's better to do, talk with the patient, an EKG every year or every month as opposed to put in a defibrillator, something like that. So I think we're not going to have a robot for every disease, all 7,000 of these diseases.
Any other thoughts?
I'm not a doctor, but I want to make an analogy with the emergency department, right? There are clinical decision support tools that are now being developed to help physicians, as you guys know, in the emergency department. They get huge amount of information that they need to act on in a very, very small period of time. So in extrapolating this concept to newborn sequencing and your question about the medical sense on what to do with the information, a point of view I get from physicians is actually maybe we don't really need to understand everything, neither to know everything. We just need to be able to act on the information. So even we cannot know what an exome is or a genome is, if we know what to do with it, that's exactly what we need.
And even where there is a solid guideline to be followed, that's not the only factor in decision making. At least in the US, there's a huge amount of defensive medicine that gets practiced, a lot of worry about not following through with something that a patient may have requested, even if it's not entirely reasonable. So, you know, there are those other, it's not all about the guideline.
Yeah. And even if you, and I keep raising this theme of, you know, is this within the public health ecosystem or is this outside the public health ecosystem? Even if you believe it is, you're flipping into the clinical ecosystem once you've identified an at-risk individual. Okay, let's open it up for questions with our mic runners out there. Please keep your questions brief, maybe just one part, not 3 or 4 parts. And we'll keep our answers brief. And so we can get a lot of questions and discussion in, please.
Thank you. My name is Shanti. I'm a metabolic geneticist based in Sydney, Australia. There's compelling benefits to newborn screening, but I think we've all acknowledged the concerns as well. I'd just like to focus on consent, and particularly from a genetic literacy perspective. In Australia, the consents are obtained by midwives. Now, there are some research projects that are labeled as whole genome sequencing. Midwives will not be able to understand that we're actually not doing the entire genome, but focusing on targeted, treatable or actionable conditions. And so there may be miscommunication of information. I've seen that in clinical practice where I've had patients and families coming to me saying that they have had whole genome when actually it's just a... So how do we ensure that correct information is being communicated?
Okay,
To
the
families?
question is focusing on consent. How do we assure that there's not misunderstanding? Responses?
Yeah, it's very difficult, right? One thing that's become clear, at least in the US, is that a huge amount of genetic services are being delivered by genetic counselors, of course, but also nurse practitioners, physician assistants, and we have a big focus on developing not only educational materials for that group of practitioners, but also materials for them to use with patients, because this is a huge hurdle for them.
And not everybody has to respond to every question, but any other responses here? Okay, great. Thank you for that question. Yes, May. Oh, you need a microphone.
There's one up there first.
Oh, sorry, one up there first. Hi, Stephen.
Hi, I'm Stephen Brenner. I'm a computational biologist at UC Berkeley, and I'm part of the UCSF project NewbieSeq. And I want to ask about a practical challenge that I haven't heard much about yet here today, which is regulation. For example, in the United States, the FDA has just issued a notice of proposed rulemaking, which will regulate laboratory-developed tests, LDTs, as devices. When the FDA originally planned to do this in the Obama administration, newborn sequencing was their first area of focus, which had serious implications. BabySeq and also my project NewbieSeq significantly curtailed study design as a result of their approach. Could you speak to the impact regulation will have on newborn sequencing projects, on innovation, and the benefits and risks that you see?
Sure, and I'll just say from a BabySeq point of view, this was a really traumatic experience that the FDA took an interest. You do not want the FDA to take an interest. I mean, most people, you say you're going to take an interest, it's great, but not them. Anyway, they took an interest in BabySeq and several of the other things, and it was horrific. It almost derailed the entire project. But it's a great point. I don't know. I think that influences everybody, including the diagnostic companies. So Paul, why don't you start us off responding to that, and really not just asking questions, but anybody here who's interfacing with any of the regulatory space, please jump in and contribute, because this is a terrifying development in the US.
Yeah. So what Stephen's alluding to is this week the FDA wants to put out a proposed rule that they're going to consider laboratory-developed tests as medical devices. So, in a sense, regulating genetic laboratories. So that's the background. We don't know a lot of the details about.
I mean, that sounds really okay, right? I mean, shouldn't they be regulated?
Yeah. Well, let's just from 60,000 feet, it's always in the best interest of the patients, which I think they should be first, in that they get a quality test for sure. I will say that laboratories are regulated right now by CLIA, CAP, and New York State. New York State has a very stringent level of certification. So how all this is going to play out, exactly what they're going to want.
So the fear is what they will require.
Yeah.
In terms of participant understanding, quality assurance, validation of individual genes and variants.
Yes.
Before they will let you do what you're doing right now anyway, right?
Correct. And so our field, genomics, is a very fluid field. There's a lot of innovation that is applied to what we do. These are new genes, new types of variants. The word AI has been bounced around a lot at this conference. So think about all those innovations. What will be the requirements to add that to the present testing are really some of the questions. We're, as a commercial lab, and I'm sure other commercial labs, we're looking at this very carefully. ACLA has come out strongly against it. They've released a public statement. So, I mean, great point.
We have people here from the American Public Health Laboratories group. Yep. And anybody want to comment from?
Has ultimate authority over CLIA and CAP and all these things. So, you have to remember that CLIA, they have ultimate authority over all this stuff. So they will determine what happens, and they have the federal law authority to do so. I mean, I think the concern is for ultra-rare diseases, you cannot have, you know, FDA, nobody's going to develop an FDA IVD kit for an ultra-rare disease that's not going to make any money. I think the FDA will leave it alone. But when it gets to newborn screening and populations, even though it's rare, the FDA is going to take it very seriously. And, you know, they should, because 23andMe is going to tell you you might develop some disease, and they better be right. So I don't know. It's a very complicated issue. But the FDA will not budge just because it's a CLIA test, right? So.
Right. Over here with May.
Yeah. May Baker, I'm from Wisconsin Newborn Screening Program. I do newborn screening every day. So the follow-up this is many years ago, when talk about LDT, newborn screening, and go back to Robert comments, this is not diagnosis laboratory. So we have to put the two groups. So that time, my understanding is newborn screening test because it's a risk assessment, and the recommendation is do confirmatory test. So they're put in the category is low risk. So I think going forward to make comments, I already get notification to make some comments. That's the principle I'm going to use. It's because it's unlike clinical diagnosis, you don't base on that to do the treatment. So I think that's could potentially beneficial for newborn screening test.
So also just following up on that, so one of the salient points is, would we consider newborn sequencing to be low risk? That's going to be a key point. So let me ask for a vote from the audience. Not that the FDA will care, but what we think. But how many people would? Okay, let's say we take the intersection of all these Venn diagrams. Let's just take a really low number. I think Zornitska's Venn diagram had 58 common genes that almost all the groups had in common. So let's just say 58 genes that every group agrees are treatable. And if we put that to the FDA, how many of you would, and you were sitting on the FDA, how many of you would say that's low risk? First of all, how many people would say low risk? And how many people, I think it's only low and high that you're allowed. There's not a medium? Is there a medium? Okay, what's it? Okay, how many would say moderate risk? And how many would say high risk? Wow, you stay away from the FDA. Okay, other comments on this? Or, yeah, Stephen, you want to circle back and we should go on to other topics, but this is such an interesting one.
High risk variants, so they're high risk.
Oh, yeah.
Robert.
Yeah, fair point, fair point.
I just want to circle back and note that the FDA's measurement of risk often differs from that used by IRBs and by researchers, and so that's important to appreciate. And second, that even when there are no real hurdles that are put in place, just the procedural processes are those which researchers especially unfamiliar with and highly burdensome, and so that's something to be bearing in mind as well.
Great.
So I was just thinking about high risk, and like for genomic sequencing, if you get a pathogenic variant, there's a very high probability that the child has the condition. It doesn't mean it's still just a screening test, and but it's a very high probability. And so the question is whether the FDA will worry about high specificity of the test, even though it's low sensitivity.
Wait, did you just say that if the child has a pathogenic variant, there's a very high probability that they'll develop the disease, that they will have the disease?
So that's the kind of one of the principles that we've been trying to focus on is very penetrant.
I'm not sure everybody would agree with that, but...
So that's the thing, is it's still a screening test. But
can we vote on that? Okay, let's move on, unless you have a final comment on this, let's move on from the regulatory question.
I just wanted to point out there is countries, including Australia, where this is already the case. So laboratory tests are regulated by, our version is a TGA, and we call it an in vitro diagnostic device, not a medical device, but ultimately it's same, and that's been in place for many years. Regulation is important, but it has to be done well. It can be very harmful if it
and as
goes
Stephen
over the
pointed
top.
out, it's not really the regulation qua regulation. It's the burden, the ambiguity, the time, the effort that it puts on people involved. Sorry.
Just one more comment that, one thing which we have not talked about is that NGS today, even today, is not a settled technology. I mean, it's dynamic. We just heard about long read. So how is FDA going to look at
Right,
that?
right.
Because
Well,
almost every lab
Let's not get into,
is, and
This could really absorb us. We'll have drinks over this. But Melissa?
I was just going to say, when you're asking about risk, I think most of us were thinking about risk to the baby and risk to the family, but I also think we need to consider risk to the healthcare system that is completely unequipped right now to handle any kind of uncertainty resulting from even a simple, straightforward kind of genomic result. So that's a relatively high risk, I think.
So from a treatment perspective, when you hear all this, does this give you pause? Should you stop working on these treatments?
No, so I don't think that we should stop working on it, but one of the issues that came up in the preliminary conversation that we had was about workforce and who's going to be managing all of these results. It's been true for a very long time that there aren't enough geneticists. There aren't going to be. There aren't enough training programs. There aren't enough applicants to those training programs. And this is really going to fan out to sometimes in the US called physician extenders, like nurse practitioners, PAs, and professionals like that. And at Sanofi, we fund fellowship programs, and it has been true for at least the last 5 years that the great majority of applications we get, we call them fellowships, but it really, just funding additional training in LSD, in our case, LSDs, or really rare disease more broadly. The great majority of those applicants are not physicians, and the awards go largely to non-physician programs. But we move forward with that because otherwise there's
Well, it is a testament to something that we are all here, and we were here many of us were here last year, and these programs are being put into place despite these somewhat intractable sounding obstacles. We do believe somehow they're going to be solved. Solved enough to save some babies on the way to being perfect. In other words, not letting the perfect get in the way of the incrementally good. What other questions for this group does anybody have?
Can I?
May, did you not get to finish your question, or did
No,
you? No,
I never get
Oh, I'm sorry. Please.
So
Apologies. Please complete it.
Okay, my question is, I think we kind of going to different, like public health and clinical diagnosis. Seems the line is very broad. But for conceptually, I think if you really is identified individual without a clinical symptom, that's a screening. You can't avoid talking, that's a population based. And I was thinking that's public health. So I think we are in this kind of new area, and maybe we should think of the box, not a truly defined, screening. Can we build up the one ecosystem? I don't have a solution, but I think in a different setting, I talk about can we working together in a way, meaningful way. I heard limited comments say, we are partner with public health. What I heard is you have access to the dry blood spots. Anything beyond that, I'm hoping to hear more.
I see.
Because,
So if I understand you, you're saying this is you want this to be firmly in the public health ecosystem, but you want more than just, piggybacking on the blood spots. Yep. Okay. And I would say that that's a very fair aspiration. There is other ways you can think of it. I mean, pediatricians do clinical screening. Aren't they supposed to do a lipid level at age 1 or something like that? They may or may not actually do it. They could do this in part of their clinical practice. That could be a very different model than a state-mandated newborn screening sequencing model. So there are other models to consider. But yes, I hear what your aspiration would be. Yes. I can't see up there.
Hi,
hi.
I'm Anna Need from Genomics England. I was very interested, Paul, when you were talking about the sort of fuzzy area between screening babies in the NICU with whole genome sequencing and screening babies prospectively for newborns for disease. I'm guessing that sometimes it happens that you find something in the baby that's going to cause very early onset treatable severe condition that's not related to why they're in the NICU. And I'm wondering, I guess, who decides whether something's an incidental finding, whether you're going to return that to the parents, and whether there's some learnings for all of us from those situations.
Yeah, so again, just to make that line, the NICU, rapid sequencing in the NICU, that's diagnosis, 100% penetrance, sick child, and the diagnostic rates are very high in the NICU. In the medical literature, between 20 and 40%, somewhere around there. I think most people would agree if you look at the larger studies. Presumably healthy babies in a newborn nursery, much lower rates, and that's where we're going to find the incomplete penetrance. We're going to find variants in genes. Is this a healthy child who is going to get symptoms later in life, a patient in waiting, or is it a variant that puts the child at risk for something?
Paul, I think she's asking about incidental finding. If you do an exome or a genome, are you identifying potentially incidental findings, and are you reporting them?
Yes, we are. That's a whole interesting, in the U.S., ACMG, the American College of Medical Genetics, has dictated that we look for incidental findings, BRCA1, BRCA2, a lot of cardiomyopathies.
Recommended, not dictated. Okay.
Yeah.
Just saying.
We have to ask the parents, do you want to do this or not? Opt in or opt out of these secondary findings. Let's just use BRCA1 or BRCA2. Imagine these are parents with a sick child. They have a lot on their mind. And that's actually a barrier to testing, is to have to consent the family to talk about BRCA1, BRCA2, other conditions that might affect this infant as a baby when the child is critically ill. So clinicians have to do that. They have to discuss secondary findings. If they opt out of it, we don't look at it.
As the guy who wrote that, it's recommended, not have to. Just keep saying. Yeah. Oh, sorry. But I want to circle back to Vanessa in just a second. But if there's a question ready up there, yes, please.
Ulrik Stolte from Denmark. I was thinking about the clinical decision making, and you're talking about how you need expert opinion. I think that's a good place to start. But in the end, we need randomized trials to see which clinical decisions do what for patients. But these are not drugs. These are more screenings like EKGs and MRI scans and so forth. And the pharmaceutical company doesn't really have any interest in developing those kind of platforms because they're not patentable. But this is the intervention that might be relevant to a lot of the diseases that we're discussing here. So where do you see the role for companies in developing that, or is that purely an academic?
And that you're saying the companies need to develop is the clinical decision support?
Yeah, or like a randomized trial of, for instance, doing annual MRIs versus not doing that, or doing it more frequently.
Let me tweak that question a little bit, and Vanessa, because it actually ties in with something I wanted to ask you. Many people would find it impractical to do large-scale randomized trials. You'll hear later about BabySeq, which is a small-scale randomized trial, but large-scale randomized trial with thousands of people not receiving or receiving a lesser product is pretty hard to pull off. And but Vanessa, one of the things I think Danaher is particularly thoughtful about is big data. And one of the dreams of big data is being able to take large-scale material. You know, Genomics England's already talking about AI, and intuit inferences, in maybe even clinical decision support inferences. Any thoughts on that from, you're embedded in this company that's really, really talking about scaling and data. Any thoughts about this?
Yes, when it comes into, again, more on the focus of the biomanufacturing for treatments for rare disorders, right? One of the areas that we are experiencing right now in the U.S., as you know, and similarly in Europe, I guess it's on the rare disorders space to start thinking, how do we take N of 1 and you make it N of 1,000? How can you go from thinking of this is one antisense oligonucleotide for each individual patient to this is a chemical class that I'm going to develop for any type of mutation, and going more towards what actually Peter Marks is calling platforming manufacturing, right? So when it comes into data, the link I want to make here is that one of the power of newborn sequencing that we see, and that's the reason why we are thinking how can we be even more active in initiatives like this one, is that with newborn sequencing, you open now the ability to find maybe a small subset of patients that have mutations that are amenable for the same type of therapeutic, let me put it this way, a therapeutic class, like antisense oligonucleotides. I know that Genomics England is doing that with some studies, the UK pilot and many others, but when it comes into data, if we are able to access the data not only on the sequencing, but also the manufacturing data and understand what really drives efficacy at the end, that's going to be the way that we can really unlock what's necessary on the manufacturing process to develop a treatment that can act on the patient. And the point is that the reason I'm making this comment is because, as you guys know, biomanufacturing, it's expensive. Every time we talk about a drug costs $300,000, it's very expensive to manufacture. Well, if we really want to take this super expensive manufacturing process to something amenable for rare disorders, we need to really understand what are the steps we need to change in the whole process to make it more effective. And that's all, honestly.
So to the questioner's point, you could think about a trial. Once you identified an at-risk individual, you could think about a trial of a new medication or a new treatment. I kind of heard your question in terms of decision support, like what do you do next? And if how do you do a randomized trial of like, should you intervene early? Should you intervene late? Should you get annual echocardiograms? Should you get monthly blood draws? I mean, the number of variables that you would play around with in a trial of downstream surveillance and intervention would just be mind-boggling. So that would be pretty intense.
Yeah, I would just comment that there's certainly a role there for registries, disease registries, many of which are industry supported. And just thinking about, for example, so much of what we know about Gaucher disease and what's useful in monitoring for Gaucher disease comes out of 30 years of registry data. And in terms of, and registries, of course, have their limitations in terms of prospective randomized trials. True, it's unlikely that pharma companies, generally would pursue those on their own, but there is a mechanism through which we can fund investigator-sponsored studies, independent studies, and we often use that mechanism. It's not the case that we can fund every single thing that comes our way, but we do get those kinds of proposals and view that as a very important mechanism. Right, exactly.
Even when they're not drug studies.
Yeah,
so that's
for
cool.
sure.
Good to know. You'll get about 100 applications tomorrow.
Love it, love it.
Okay, we should wrap up here, but I'd like to ask each panel member to give about 30 seconds, but we'll take one last question. Yep. And then, but while you're thinking of your last 30-second statement you'd like to make to the group.
Maybe to clean up a few things. It is not true that if you have 2 pathogenic mutations that you always have a high probability of getting the disease. Just look at Pompe disease where it is not true. So all eyes are on Genomics England. If you have to focus on highly penetrant, clear-cut, serious genotypes.
Is there a question here?
Then the FDA will be okay because you do minimum harm. But if you don't do that, it's going to implode downstream biotechnology, family follow-up, and the FDA is going to be all over you. Okay?
Okay, thank you.
I think it's clear.
All right. I think we've got lots to find out in the arena of penetrance, and it's going to be very exciting to see all the groups that are able to follow. Are able to follow the clinical follow-up. And not just what's in the medical record, right? Our group has looked at biobanks, pulled people out by variants, who were never identified with the disease, and yes, they have the disease. They have features of the disease. They have clinical features of the disease. So it's not just enough to do an electronic survey of the EHR and say, it's not penetrant. They're out there. Okay, last 30 seconds. Who'd like to go first? Final statements. Paul?
All right, yeah. Two things I want to say is, first of all, these projects that are being presented are research projects, and there's so many questions to be answered, and the idea is that we're gonna figure this out, and so that's the one thought, and the second thought is the power of patient advocacy groups in the U.S., at least in North America, are an incredible force. They have, as Don Bailey said, I'm going to steal your phrase, fire in their belly or fire in their gut. And I think they're going to play an important part.
Thank you. Either one of you?
I would say, I'm sure you guys are going to figure out all the diagnostic component, but it's important as well to think about it's the access from educating the physician community, but also access thinking about how do we develop then treatments for those patients.
Thank you. Final statement?
I would just say there was a very important conversation earlier about the meeting that happened at the Liberty Fund, and I was thinking as that was being presented that pharma companies are often called on to fund some of these projects, and again, we can't and don't fund every single thing that comes our way, and without a nuanced understanding of that kind of conversation, we are then using our own biases and maybe unwittingly steering things in a certain direction that we don't mean to, not being aware of that conversation.
Right. And I'll just wrap up by saying that we have an opportunity to look in multiple directions at the upstream and downstream, but I'm not sure it's our responsibility to solve the inequities of healthcare writ large. And we have to play the cards we're dealt when it comes to, at least in the United States, a fairly dysfunctional healthcare system. So our question really isn't, how do we design this so that it's perfect? Our question is, how do we design this so that it can work for some, prove out its benefit, and grow? I think it's important to keep that in mind. Thank you so much to our panelists. Let's give them a big round of applause. And thank you for the opportunity to chat.